Start.
Yes, dear students, in the bioinorganic chemistry, we are left with two important short answers. One is carboxy peptidase A, and another one is carbonic anhydrase. These are the two enzymes. So, in which we have main metal is zinc, whose oxidation state is +2. So, before studying these two enzymes, let us have some information so that we can easily understand these two enzymes. One is, we must know what are these proteins. So, proteins are generally made up of amino acids. Amino acid means it will have one amino group, CH or COH. So this is acid group and this is amino. So amino acid. We have studied in proteins, that is in amino acids and proteins chapter. We have learned different varieties of these amino acids. Suppose if R is replaced by H, then it is called glycine, GLYCAN. So generally a three-letter symbol may be typical for these amino acids. So glycine, suppose if I write CH3, then it will become alanine. So ALA. Like that we have valine and leucine. So these four are there in our syllabus. You have already learned in amino acids and proteins in that unit. Now here, let us have how these amino acids they will combine. This is one amino acid. I am writing its general formula. Alkyl R. R may vary. It must have two functional groups, that is amino group and carboxylic acid. Now I am taking another amino acid, say H, NH, means it is nothing but NH2. So then we will write CH or alkyl group, then COH. This OH and one hydrogen from another amino acid minus H2. Then we get NH2, then CH, R. Then we have CO, COO. Then we have NH, okay. Then CH, R, then COOH. So like that, we get a dipeptide. Here, two amino acids, they will combine and they are using one water molecule and it is giving it a dipeptide like this. This is called dipeptide. Okay. So here a large number of such amino acids, they link with one another. Here you must remember this bond, CONH bond, that is called as a peptide bond. Peptide bond. Bond is also called linkage also. So peptide linkage also we can call. So like that, a large number of amino acids, they will combine with one another, and by means of these peptide bonds, they will form a long chain. So here I will write a general formula of this protein like this. First it must have one amino group, then CH, then R, CO, then it will be linked with another amino acid, CO, which is a peptide bond. So here we have CH, then R, then CO, NH, then CH, R, CO, then NH, CH, R, C, let us take COH. Now here I have taken 3 amino acids. Now R is varying, R is different. So here how many R's? 4. That means 4 amino acids are linked. Now instead of this one, let me write GL1, that is glycine. Let us replace this one CH by alanine AN. Replace another one. Say this is a valine, and another one is say leucine, like that. So when these amino acids, when they combine with one another by losing one water molecule, they are getting linked with this CONH, peptide bond or peptide linkage. In this case, generally, always we represent this amino group on left hand side and carboxylic acid group on the right hand side. So generally, this carboxylic acid terminal, that is called as a carbon terminal, and this one is called as a nitrogen terminal. So in any amino acid we have NH2, then CH, then R, COH. So amino acid is having 2 terminals. One is a carbon terminal and another one is a nitrogen terminal. So while writing the structure, you must depict or you must show the amino acid in such a fashion that amine group is always present on the left hand side and the COOH group is present on the right hand side, even in the case of a protein. Okay, that you have to do. Next one, in the case of nomenclature already you have learnt, we must write the name of these amino acids in sequence. You should not change the sequence. And are there any changes in the total name of this polypeptide? Poly means many amino acids are linked with peptide bond. So here in this case, the carbon terminal possessing amino acid whose name does not change. There is no change in the name of the carbon terminal amino acid. That means the name of this polypeptide will end with leucine, Leucine. But the remaining amino acids, whether it is the first one or the next one, we must remove IN by YN. So here first amino acid is a glycine. So GLYCINE. So remove this IN and add YN. That is the rule. So then I will write, keeping this in mind, I will write this glycine. Why? Because I have replaced INE with Y. Then this is alanine. So alanine, MYL. Then valine. So VALI. Valine means LIN. So valine. The last one is leucine. LEUCINE. So this is the name of this polypeptide. So glycine, alanine, valine, leucine. Okay. So here no need to write the names of these proteins, not necessary. So just in order to have what these enzymes are doing here, actually carboxy peptidase A, this enzyme is connected with this protein reaction, that is hydrolysis. So when a protein undergoes hydrolysis, what happens? This reaction is catalyzed by carboxy peptidase A. It will shorten the length of the protein. That means it will break the large sized protein into a small sized protein. So where the protein is undergoing a cleavage break. So cleavage means break. Generally it undergoes cleavage at the carbon terminal. At the carbon terminal only the cleavage is occurring. Okay. Now this is the information we need for understanding this one. Next I am moving to. So no need to write all this information in the examination. For having proper understanding of the function of this carboxy peptidase, I have given this protein information. We will write here in the function. What is the function of this carboxy peptidase here? You will write hydrolysis of a protein. A cleavage occurs at the carbon terminal, and the size of the protein is shortened. That means size is or the length is reduced, decreased, like that. Now I am moving to the second one, that is carbonic anhydrase. So this is also another important enzyme in which also zinc is present. It is concerned with the buffer action. So first of all we must know what is a buffer solution. Buffer solution. A buffer solution is a solution that contains a weak either acid or a base. So weak acid plus its salt, or weak base and its salt. Now I am taking an example that so that it will be clear. CH3 COOH. This is acetic acid. So this acetic acid is a weak acid, and I am taking its salt. Replace this H with Na, then we get CH3 COOH. So sodium acetate. So this is a salt. Now this buffer solution, it is called as an acidic buffer solution. Like that we have basic buffer solution also. What we need here, weak base. Say for example I have taken here ammonium hydroxide plus its salt. I am taking ammonium chloride. So this is a weak base and this is a salt. So here I have used a base and its salt. So this is called basic buffer solution. So a solution which contains a weak acid and its salt, or a weak base and its salt, and its job is, what is its main function? It will maintain constant pH. For maintaining this pH of a solution, that is hydrogen concentration, logarithm of hydrogen concentration is called the pH. So no need to write the definition, not necessary. We want here blood. It is having a pH range of 7.35 to 7.45. This is the pH range of blood. This must be maintained constant. So this function was done by this carbonic anhydrase. How? That we will study later. So this is the information here we want. Actually in the case of human beings, in human physiology, we have buffer systems. 3 types of buffer systems are there. So all this information no need to write in examination. Buffer systems, in this case, one is carbonic acid. Carbonic acid or bicarbonate. Bicarbonate buffer system. This is one type. Second one is protein buffer system. And third one, last one, is phosphate buffer system. So like that here, we have three varieties of buffer systems in the human physiology. Buffer systems. So what is its main function? It keeps up the pH of blood. This one, carbonic and bicarbonate buffer system. It is concerned with the maintenance of pH of blood. That is having a range of 7.35 to 7.44. In the case of a person who is having a defect, suppose if it is less than 7.35, then the defect is called acidosis. If it is greater than 7.45, that is called alkalosis. So this is extra information. So like that, first enzyme, it is concerned with the digestion, and here its main function is hydrolysis of protein. That you must know. Now I am directly moving to the two enzymes. So this is the information which we bear in mind for understanding these two enzymes. So both enzymes, they are zinc metalloenzymes. That is also a zinc metalloenzyme. In this case, the oxidation state of zinc is +2. Here also it is zinc +2. And so zinc is the center of coordination. So in the case of a complex, zinc is the central ion in both carboxy peptidase A and carbonic anhydrase. Both are having zinc in it. Now let me write what is the valence. In the case of carboxy peptidase and in the case of carbonic anhydrase, both valencies are 4. Here also the valency is 4. That means how many ligands are forming a coordinate covalent bond with this zinc metal ion? Now let us have this one, please. Zn plus 2. You can imagine the form of a tetrahedral structure. One valency, second valency, third valency, and fourth. Okay. Here also, I will draw the same bag, Zn plus 2. One, two, this is 3, and this is 4. Now, out of these 4 coordination sites, you can call like this also, coordination sites. Yes, site means regions, 4 sites. Okay, this is it. In the case of carbonic anhydrase, one amino acid by name histidine. 3 histidine amino acids, they will link with zinc metal ion. How? With one atom, is donor atom. No need to represent the donor atom. Here you just write HIS. Here also one HIS. Here also one histidine. HIS. So with the help of a donor atom, 3 histidine amino acids, they will occupy the 3 coordination sites of this zinc. Whereas in the case of carboxy peptidase A. One is glutamine, glutamine. Glutamine. One is his, that is histidine. Here also histidine. So only two histidine amino acids and one glutamine amino acid, they will link with the zinc ion with their lunar atoms. Fourth coordination site is always empty. Here in this case, say for example, in the case of carboxy peptidase A, during hydrolysis, water molecule will occupy that fourth site. So here we get the H2O, H and H. Oxygen is having two LPs. So one LP is given to this like that. The coordinate covalent bodies of water. So here also during the buffer reaction, here we have H2. Like that you have to do. Now the most important point, the first one, make it your first point here. Carboxy peptidase A, it is having 307 amino acids. So total amino acids present in carboxy peptidase A is 307 amino acids. They are linked with peptide linkage. And what is this molecular weight? The molecular weight of carboxy peptidase is 34,600. What about the carbonic acid? Carbonic anhydrase. This is having 259 amino acids and its molecular weight 30,000. Okay. So carboxy peptidase A and carbonic anhydrase both are zinc metalloenzymes. That means zinc metal ion containing enzymes. In the case of carboxy peptidase A, it is having 307 amino acids and its molecular weight is 34,600. Whereas in the case of carbonic anhydrase, it is having 259 amino acids and its molecular weight is 30,000. Okay. Now I am moving to its function. So regarding the function, what is the function of carboxy peptidase A? Means hydrolysis of protein. Okay. Then cleavage occurs at carbon terminal and length of protein is decreased, that is shortened. This we have to write when we get carboxy peptidase A short answer. And here we represent the protein families. Now NH2, I will take glycine, then it must have peptide bond, NH, then I am taking alanine, alanine, then CONH peptide bond, then valine, C double bond O, NH, then Lucy, NE, CO, NH. Let us make this one COH, COOH. So here I have taken 1, 2, 3, 4. 4 amino acids. Generally cleavage break is occurring at carbon terminal CO double bond. So this structure is like this is COOH. So break occurs only at the carbon terminal. So suppose I will break here. This is the carbon terminal. Then what we are getting here? Gy and A. So glycyl alanine. I told you the amino acid which is containing COOH group, in its name there should not be any change. You should not replace INE with YN. That's why it will become glycyl alanine. Now what about this one? Here we have valine. So valine and leucine. This much you can write in the examination. So a breakage, cleavage is occurring at the carbon terminal. That you must write. That is the most important point. So this is the information regarding carboxy peptidase E. Now I am moving to the next one. What is this carbonic anhydrase? Carbonic anhydrase is a zinc metalloenzyme. It is having 259 amino acids. Its molecular weight is 30,000, and it is doing, it is catalyzing the buffer reaction. Which buffer reaction is carrying out? Generally, HCO2. This is bicarbonate ion. Phos H positive gives rise to carbonic acid HCO2. CO2 phos H2. So this reaction is catalyzed by carbonic anhydrase. That is bicarbonate ion. It will combine with hydrogen cation and it is forming carbonic acid. So this is carbonic acid. This is bicarbonate ion. Bicarbonate ion is converted into carbonic acid, and carbonic acid is converted into carbon dioxide and H2. And this CO2, it is exhaled from lungs. Now I will do the link. How it will happen? So carbon dioxide being exhaled from our nose, that is from the lungs. Generally, hemoglobin, it will combine with the oxygen and it will become oxyhemoglobin. That we have already learned in your URU or biology classes. So this hemoglobin will supply oxygen to each and every cell. In the cell, cellular respiration. So cellular respiration. So what is the reaction that is occurring in this cellular respiration? In the cellular respiration, say if you have glucose, it will burn with the oxygen. So burning of any material with oxygen is called as a combustion. So glucose that is present in the cell, it will combine with oxygen undergoes combustion and it will form, it will release energy. We call it as adenosine triphosphate ATP and CO2 and water. So like this, the glucose, the sugar that is present in the cells it will undergo combustion and it is releasing the energy and the carbon dioxide and water. This carbon dioxide, it will dissolve in blood, and in the blood what happens, that the carbon dioxide is converted into carbonic acid. This carbonic acid is split up into bicarbonate and phosphate. So this bicarbonate, it will flow in the blood circulation and it will reach the lungs. At the lungs what happens? Once again bicarbonate will combine with the hydrogen cation and it will form carbonic acid. Then it is converted into carbon dioxide and water, and carbon dioxide is expelled out through the nostrils. Like so, we must have these two reactions. Cellular respiration is responsible for release of carbon dioxide. How it is coming out? Because of this buffer reaction. The total buffer reaction here have is bicarbonate and hydrogen cation gives rise to carbonic acid, and carbonic acid is converted into CO2 and H2 and it is used to us. So this is the main function of carbonic acid. Because of this buffer reaction, the pH range, so pH range of blood will be 7.35 to 7.25. That is required for normal functioning of any reaction, any biochemical processes that are occurring in the human. So it is human. Like that, blood pH is maintained.